This is a Fauzaan's IB video on the light-dependent reaction. The IB has a lot of understandings when it comes to the light-dependent reaction. However, these essentially just boil down to the different steps and the different processes that we will discuss throughout this video. So to explain the light-dependent reaction, I'm going to be using two different diagrams. Both are from the high-level biology textbook. Now, at first they may appear quite complicated. However, once you break them down, they are fairly simple to understand and pick up. So this is the first diagram, which contains three of the four processes. Process number 1 occurs here. Process number 2 is this photosystem 2, or P680, which is the entirety of this section here. Photosystem 1, which is section 3, is again this entire section here. And then finally we have this process, which is known as chemiosmosis. So the four stages of the light-dependent reaction are photolysis, photosystem 2, photosystem 1, and chemiosmosis. Now, the light-dependent reaction occurs in the intermembrane space of the thylakoids. So if we look at the chloroplast over here, we can see that you have a granum of several thylakoids, which is just a stack, and a thylakoid is a singular one. Okay, so if we are to break down the stages of the LDR, we're going to first look at photolysis. So photolysis occurs over here with this water splitting enzyme. So if we isolate this bit of the diagram, we can attempt to understand this process a lot easier. So photolysis is the splitting of water using this water splitting enzyme to split water into 2 hydrogen ions and half an oxygen. Now, this process also produces electrons, which are donated to photosystem 2, which is the second part of the light-dependent reaction. Now, this entire process of the water splitting enzyme is derived by light photons, hence why this is called the light-dependent reaction. It will not occur in the absence of light. So, using light photons, the water splitting enzyme splits water into hydrogen, 2 hydrogen ions, half an oxygen, and electrons. And the electrons are donated to photosystem 2, which is also known as P680. And then once again, photons boost the energy level of these electrons. And as a result, we start the hydrogen ions help to develop a hydrogen ion gradient. This will make a lot more sense later. And the oxygen that is produced is released from the stomata. So if we once again look back at this diagram, we've now somewhat explained what happens in this section here. So the electrons have now made it into photosystem, have now been donated to photosystem 2. So if we were to have a look at photosystem 2 in more detail, the high energy electrons just gain their high energy state from light photons are accepted by this excited electron acceptor over here. Now this electron acceptor passes the electrons onto the an electron carrier known as PQ. This causes the electrons to be delivered through an electron transport chain. So I just abbreviated it here. An electron transport chain to the cytochrome complex, this electron carrier. Now, what the excess, because obviously these excited electrons are high in energy, the excess energy that they have is utilized to pump hydrogen ions across the membrane in the cytochrome complex. And this once again also helps setting up an hydrogen ion gradient. So now we have established that the water splitting enzyme produces electrons that are accepted by the high energy electron acceptor in photosystem 2. They are carried by an electron carrier PQ through the electron transport chain, and as a result use their excess energy to pump hydrogen ions across the cytochrome complex, once again also aiding in making a hydrogen ion gradient. So now the electrons have come through there, through the electron carrier. They now end up at photosystem 1. Now, once the less excited electrons arrive, they are once again, at this point, they are excited by photons of light, and once again accepted by excited electron acceptors. Now, this time the once again an electron carrier, this time known as ferredoxin, transports them through the electron transport chain. This electron transport chain contains NADP reductase. Now, what NADP reductase does is it reduces NAD plus in the area and combining it with H plus ions to produce NADPH. So, to somewhat recap once again, the water splitting enzyme produces electrons. The electrons are accepted by the excited electron acceptor in photosystem 2. The electrons are transferred using the electron carrier PQ through the cytochrome complex, which pumps hydrogen ions across the membrane, setting up a hydrogen ion gradient. The low energy electrons arrive at photosystem 1, where they are once again excited by photons and once again accepted by an excited electron acceptor. This time the electron carrier ferredoxin transfers them through another electron transport chain, this time containing NADP reductase, which synthesizes NADPH from NADP+. Now, this is another view of the light-dependent reaction. So we can observe photosystem 2 here, going across the membrane. We have this cytochrome complex where the electron carrier PQ takes the electrons in the electron transport chain through the cytochrome complex, pumping hydrogen ions across the membrane. However, now we can observe this entity known as ATP synthase. Now, this final process is known as chemiosmosis. So a strong hydrogen ion gradient has already been created due to the fact that the photolysis of water has produced hydrogen ions, as well as the fact that the cytochrome complex has pumped more hydrogen ions into this thylakoid space using the energy from the electron carrier PQ. So now these H plus ions now move passively back through the membrane through ATP synthase, and what this does is it allows the phosphorylation of ADP to ATP. So as a result, if we look at the general equation of photosynthesis here, the reactants used in the light-dependent reaction, it is observed that water is used because of the water-splitting enzyme in photolysis. So and the products we produced are NADPH, ATP, both of which will end up going on to light-independent reaction, which is a reaction that occurs in the absence of light, as well as oxygen. And the oxygen can either be used in respiration, or it is most likely released from the stomata of the plant. To summarize, the light-dependent reaction occurs in the presence of light. The photolysis of water involves a water-splitting enzyme which uses light photons to split water to produce electrons, hydrogen ions, and oxygen. The electrons are then delivered to the photosystem 2, P680. The hydrogen ions help to set up a hydrogen ion gradient, and the oxygen is released from the plant stomata. The excited higher energy electrons are then accepted by an electron acceptor, which passes them on to the electron carrier PQ. PQ delivers them through an electron transport chain to the cytochrome complex, where the excess energy pumps hydrogen ions across the membrane, setting up an ion gradient. So the photolysis of water, as well as the cytochrome complex, both help to set up a hydrogen ion gradient in the thylakoid space. Now, the lower level excited electrons arrive at the photosystem 1. Photons, also known as light energy, excites them again, increasing their energy. The excited electrons are accepted, let's just say accepted, by an electron acceptor which passes them on. The electron carrier ferredoxin now transports them through another electron transport chain containing NADP reductase. This synthesizes NADPH, another product to be passed on to the light independent reaction. And the strong hydrogen ion concentration gradient is set up by photolysis and the cytochrome complex. These H plus ions then move passively back through ATP synthase to phosphorylate ADP to ATP. ATP is a product of the light-dependent reaction that will be transferred to the light-independent reaction. So once again, we have the light-independent reaction to which ATP and NADPH are transferred to. And that is the light-dependent reaction. Thank you for watching.